So you said this is the last one of the series for this?
This is the last instructional session. So being digital, we're experimenting a little bit. And so it's live and it's hands-on, and we sent all the materials to the teachers, equipment, reagents. We were just talking before you came on because there was one thing we lacked, and we're sending out, we've had a couple of weeks of sending a few straggling items. And then next week is our final session of the series where the teachers will get to do some planning. So taking a look at their current curriculum and seeing where some of these techniques and concepts they've learned would maybe be able to replace some of the current techniques that they're using. For example, now that they have access to equipment from our equipment depot, or maybe they're able to purchase from Mini PCR, they'll be able to do something more hands-on than before. Is the hope. Hello. All right.
Hello. Is that Albany? Is in Albany, New York, or Albany? I see Tia's shirt.
Albany, Georgia.
Oh, Albany, Georgia, because I grew up near Albany, New York. So.
Oh, okay.
I saw that on your resume. I'm a Rochester girl.
Oh, okay. All right. So you know about winter.
Yeah, that's why I'm here.
Yes.
Yes, absolutely. So in just respect of everybody's time, we will get rolling. So if everybody who's on here would go ahead and mute, and as soon as I finish introducing Dr. McKinley, I will do that as well. Any questions that you guys have throughout this, you're welcome to throw into the chat. And Dr. McKinley, I will just monitor the chat in case you don't see a question, and then we can talk about that. Would you prefer to wait till the end, or as you're going through for questions?
I think as we go through is fine. Yeah.
Okay. All right. Well, perfect. So, so Dr. Mark McKinley is joining us from the Task Force for Global Health, which is located in Decatur. And we have such a phenomenal public health, strong public health presence in Georgia, between the Task Force.
It's the center of the global health universe. It really is.
Mm-hmm. So we're really grateful to have you here speaking with all of us today. Whoops. So Dr. McKinley has an incredible history, and it is so complex. I have to just have these notes in front of me, but he has co-founded and led R&D at two biotechs. One is Vero Pharma Incorporated, and that was actually acquired by Shire, which is now Takeda, in 2014. And then Tetralogic Pharmaceuticals, which is currently in action and is on the NASDAQ. He's led R&D programs in antiviral, antibacterial, and oncology. So we've been learning about both antiviral and antibacterial in this training, and how to detect viruses and bacteria. While he was a co-founder and VP of R&D at Vero Pharma Incorporated, he built and led fully integrated preclinical and clinical development and regulatory affairs for organizations of more than 100 people that enrolled over 5,000 patients in over 80 clinical trials. He played a significant role in the licensing of pleconaril, which is one of Sanofi's drugs, and Vanconin. I may be butchering these names, so please feel free to correct me, which is one of Eli Lilly's drugs. He's been involved in over $500 million in venture fundraising to get these R&Ds through trial.
Just froze there, Kristen.
Oh no, it might be Kristen's daily boot that happens. I am not sure where else she was going to go.
Well,
with that,
so I'm going to pretty much cover my.
Yeah, we'll go ahead and
that
let
was
you
the
get
idea
started.
too. So why don't I just jump in? And by the way, please call me Mark. And it's real pleasure to talk with you all here. And let me just make sure I can do this here. So, yep, here we go. And okay. Okay, can everyone see that?
Yep.
Okay,
Yep.
and it's full screen, is that correct?
Yes.
Great, all
Bingo.
right, great. All right, so, and again, anybody has a question, just let me know as we go through. Oh, Kristen's back. Yeah, you froze there. Oh, no, she looks like she's still frozen. Okay.
Oh, it might just be
Right.
having issues.
Okay, so, as I thought about this talk and thought about, and this question's come up before, people say, well, how did you ever get interested in what you're doing? And it really all starts with curiosity, as Calvin says. And so back when I was 7 years old, I told my parents I wanted a microscope. And they said, what? Why a microscope? I said, well, I want to see things that I can't see with my eyes. So I went out and sold Christmas cards and got a microscope, and then went down to my grandfather's pond and got pond scum. So my mother loved the fact that all around my room I had little jars of pond scum, and I would take the pond scum and just love watching the paramecia scooting around in there. So I was always interested in things that you couldn't see with your eye. But didn't really know much about viruses or anything, obviously, till later on in my career. But really, this is the thing, and I'm sure you see it with your kids in your class, the ones who really have a curiosity for science. So then as I moved into graduate school, and really was graduate school, because I started, even though I started in the lab, which was more genetics, when I took my first class in microbiology, it really just fascinated me how something you couldn't see like vaccinia virus could cause such devastation, change the world, and just how that could possibly happen. So, what goes on with a virus? And of course, talk about that nowadays, it's even more amazing to live in something that only happens every century right now, caused by a virus. So after my undergrad in grad school, I worked on genetics and really herpes virus genetics. And back then herpes was being considered as the cause of cervical cancer. Obviously we learned it was HPV not much after that. But so I did a little work with in genetics there, and then went to Johns Hopkins School of Public Health in the Division of Biophysics. And so I was up in the laboratory, but interesting, I never thought I would ever get into public health, but I would go to lectures by people who were involved in smallpox eradication, et cetera, and thought, someday maybe I might get a chance to do that because this really sounds interesting. So after Hopkins, I went to what's called Sterling Drug, which is in Rensselaer outside of Albany in New York, and worked in a laboratory, a microbiology drug discovery laboratory. And this company actually was bought by Kodak. So Kristen, you know Kodak well, if you're from Rochester. Because they thought they had all these great chemicals for film, and they might make great drugs. Of course, they were just super toxic substances. They ultimately found out, and they sold our company to what's Sanofi now. So anyway, I started there, and I remember talking to my boss the first day, and he said, Mark, you have 2 new technicians. Why don't you go back there and find a cure for the common cold? And I remember coming home to my wife and saying, that's all they want, just a cure for the common cold. That's all I have to do. So we ended up, these 2 really ambitious folks and myself, working with the chemist and screening the thousands of compounds in the library at Sterling. We screened against a whole panel. There are 100 different rhinoviruses. So we had like a a pyramid. If it works against this one, then we test against these, and then we test against a broader selection, and came up with this compound, which was broad spectrum against a whole family of these picornaviruses, pico small RNA viruses. Oops, sorry, my cow's moving here. Sorry, that's my phone. And so, yeah, so it was really exciting to discover a drug that worked like this. It prevented mice from developing polio, paralytic lethal disease. It was really, really exciting. So I was totally hooked on drug discovery. And along the way, we optimized the compound to find, compound that was much better against rhinoviruses than it was against polioviruses. And at that point, this world famous crystallographer at Purdue called Michael Rossman called and said, hey, I've just solved the structure of rhinovirus. Would you like to see how your drug works? Because we knew it bound to the virus, but we had no idea how. So we set up this collaboration with him and published this paper, which ended up on the cover of Science, one of the high points for my career. And it was just an amazing collaboration because he helped us drug design. So shown here is a cutaway view. If you imagine a rhinovirus or a picornavirus, it's like a sphere. And so now we're looking at it cut in half. And the drug shown here, this hydrophobic molecule, is bound in the outer coat. And when it binds in the outer coat, it is like a lock in a Rubik's Cube. It basically keeps the virus from not being able to undergo conformational change to release the RNA to start the infection. So it keeps the virus intact, and it's called an uncoating inhibitor. So that was, super exciting. We were at the leading compound in the clinic for the common cold at that time. So we then, as I mentioned, Kodak bought us, Kodak sold us. And then when we were sold, I went to my boss at Sanofi, the head of R&D, and said, hey, I know you want me to stay, but I really like to leave. And could you give me a severance? Because I want to go help start a company. He said, boy, you got chutzpah. And I said, yeah. And and I also want to license my drug. Oh, really? So it sounds fast, but it took about a year for that to happen. We licensed the drug and with ViraPharma. And all I can say is, make a long story short, our stock went from less than $1 to over $100 and back down to less than $1. And it was, a real series of ups and downs. But along one of the ups was we did 2 huge clinical trials with over 2,000 subjects and showed that pleconazole, this drug for the rhinovirus cold, reduced symptoms within 24 hours in culture-proven cold, people with rhinovirus cold, reduced the duration of the cold, reduced the amount of virus excreted, so potentially how much you potentially would be infectious, and in our opinion, was safe and well tolerated. So we thought we're off to the races, fantastic. We passed this huge hurdle, millions of dollars study, but then we went in front of the FDA's advisory committee and they shot it down. And shot it down for reasons we could talk about for days, but basically they felt that there were some side effects there. This is just a common cold. We can't tolerate anything. And there was the potential, which ended up being totally bogus, that maybe it was causing unwanted pregnancies and affecting birth control, which it clearly didn't. But the bottom line was, at that point in time, the FDA said no. And we then said, okay, we would try developing it as, instead of an oral pill, as a nasal spray. We did a challenge study and volunteers showed it worked, but our investor says, get out of here. We're tired of you. And so that's when basically I left the company. So one of the things I know you all are interested in, and let me just be mindful of the time here, is what are the opportunities for people interested in science and pharma? And they're just everything you can imagine, depending on the kinds of things you like to do. Do you like being in the lab? If so, it's pharmacology, you know, my friends in CNS pharmacology, cardiovascular pharmacology, endocrinology, and our group in microbiology, both antibacterial, antiviral. Then the medicinal chemists, and the relationship between you and the medicinal chemists is like so close if you're going to be successful. And they make all the compounds, and when you find an active compound, they help, you know, really design and make better compounds. The analytical chemists who do, you know, the testing to see how pure the compounds are, and also they're involved all the way through the development phase. Then drug development, you've got the toxicology studies, how the drug is metabolized, and this is all fascinating stuff to learn. And that was one of the things our drug did. It actually inhibited metabolism. So that's the reason why they thought it would affect the birth control pill. There was formulation. We had a lot of interesting work around formulation. The part that I loved was clinical research, designing these trials with the volunteers and in individuals who have disease. Medical writing, getting it down in a succinct, clear way that can be communicated both in the scientific literature, but more importantly to regulatory agencies. Managing the projects. This is something I ended up going into in my later part of my time at the drug company, helping coordinate all the activities and develop the strategy to get something through to approval. Regulatory affairs. At the end of the day, like we learned, if you don't get the drug approved, nice job, good science, but you know, you didn't do it. And then chemical development after, you know, scaling up and being, and you've seen all this now with what's going on with COVID vaccines, how important it is to scale things up to be ready. And then once you're marketing, in order to be a sales rep, you need to understand the drug to go and explain it to doctors. So these are a whole range here of possibilities for people that are interested in science. So then, how did I move from there to the public health area? So it turns out here's poliovirus on the left and rhinovirus on the right, and you can see how similar they look. I mean, to the untrained eye, they look the same. They're both members of this picornavirus family, and they cause a range of diseases from myocarditis, paralytic polio, neonatal sepsis. In fact, pleconaril is currently we had done a trial with the NIH before I left Biopharma, and it showed it reduced mortality of enteroviral sepsis by 50%. So the fellow who licensed the drug is trying to get it approved. I'm still advising him, unpaid advisor, because I still obviously want to see this drug approved. But again, these compounds have activity against both rhino and polio. So polio, of course, you know, was obviously in the early 50s was something everyone feared. Struck suddenly, so many people ended up in these iron lungs or paralyzed or for life. And once the vaccine came out, you can see the lines here, people lined up forever to try to get vaccinated because they wanted to see, didn't want anybody to become infected with polio. And 1 in 200 people that become infected can lead to paralysis, and 5 to 10% of those end up dying. So WHO decided in 1988 that, look, we've got 350,000 cases of paralytic polio in the world in 125 countries. We need to do something about this because it's killing so many, it's just devastating. So they said we need to eradicate this disease. This would be the second disease to be eradicated after smallpox. And so the work got started with a lot of help from the Gates Foundation, Rotary, et cetera. And you can see a lot of progress has been made. There are 3 different serotypes of polio. 2 of them have been eradicated from the planet. There's polio 1, is the only one left. There were 176 cases of polio in 2019. I have to say that unfortunately, because of COVID and inability to vaccinate, this has gone up a bit this year. So one of the vaccines is inactivated. That's the Salk vaccine. The live vaccine, the little drops that you know, is the Sabin vaccine. It's a live virus. It's a live polio. It's been passed in culture so that it's attenuated, doesn't cause paralysis, but it can revert back to a neurovirulent form. And one of the things that was found out that in individuals who had B cell deficiencies, they didn't make antibodies, if they get polio or this oral live vaccine, it can continue to replicate in their gut because they can't clear it. And it reverts back to the paralytic form, can paralyze them, but more importantly, can infect other people and cause paralysis. So one of the things, I was invited to the National Academy of Sciences to talk with WHO, CDC, et cetera. Should we have a program to find antivirals to treat these immune deficient excretors? And the answer was yes. And because of that, I was asked to join a steering team at the Task Force for Global Health. So I would go down there twice a year. And that was an amazing experience. But in 2012, when my last company working in cancer stopped its R&D and moved into the clinic, I was looking for what I wanted to do next. I had an opportunity to go to Rice University, help them start new companies around their technology. And the CEO of the task force says, hey, why don't you come here and build a center for vaccine equity? So the Task Force for Global Health is not known because our founder, Bill Foege, who was the leader of the eradication of smallpox and also former CDC director, and as you can see, obviously very well known, he started the task force around trying to increase vaccination rates in Africa. And since then, the program has grown to disease elimination programs like river blindness elimination, blinding trachoma. We work with partners in over 150 countries. We work on disease elimination, and I'll talk more about that, but neglected tropical diseases, like I say, river blindness and blinding trachoma. We work in strengthening health systems, so helping developing countries get in place the kinds of systems that can help them prevent disease. And then the group that I was asked to build to ensure vaccine equitable access. The task force for its work won the Hilton Prize a few years ago, so that was a really big honor for us. So the task force is about 200 people now. When I joined, it was 60 in 2012. So just to show you the growth has been enormous. And it's an incredible group of people right there on West Ponce in Decatur. So our Center of Vaccine Equity, which started with just myself and one other person working on a small polio antiviral program, is now expanded to vaccination for polio. We have a polio surge team, hopefully can get back into Africa soon to help start vaccinating again, because there are a number of vaccine-derived polio outbreaks there. Hepatitis elimination. The former head of hepatitis at CDC is now at the task force heading this hepatitis elimination program. Vaccine safety. So one of the safety people from CDC is now heading a program and is now co-lead of the COVID vaccine safety group at CEPI. So we're very much involved with the whole COVID vaccine safety things, as well as other vaccines for epidemic pandemic diseases. We have a program called Voices for Vaccines to increase understanding so parents can make an informed decision about vaccinating their children, and also moving that into adults. And a program that's really grown, a very novel program, is in introducing influenza vaccination programs into countries that don't have them. So the task force, I help them get donated vaccines from companies, and we help these countries introduce the vaccine. It's shocking that over 50% of the countries don't have influenza vaccine even possible. So this is important to help them protect against the next flu pandemic. But now, of course, we're using everything we've learned, and we just submitted an application to CDC where hopefully we're going to be helping countries introduce the COVID vaccine, 50 countries. Hopefully we'll get started on this early in the year. So lots of exciting things going on in our center. And just to kind of end is to show you the opportunities in public health. So the program directors or program leaders, there's everything from drug and vaccine delivery programs. We have a number of people who study where diseases come from, how to manage them, epidemiologists, statisticians, people that understand how to manage data and put it in forms that can be easily understood by governments and government officials. Project management is key. Supply chain management. We have millions of doses of azithromycin or Zithromax that's used for blinding trachoma that Pfizer gives us that we have to get to countries, as well as Merck gives us the drug Mectizan for river blindness, and all that has to be managed with our folks. Public health advisors are folks that do everything. They're non-MDs, but they help with all the different aspects of from project management to implementation. And then communications. I'm learning more and more how important a good scientific communication person is for getting the message to the Minister of Finance. It's not the Minister of Health I need to convince, it's the Minister of Finance. And because they're not going to part with any money for anything until you show them it's worth it, their time and money. So last but not least, I just want to, oh man, I got 2 minutes ago, is just to say there's nothing like working in public health. This meeting in Bangkok a few years ago, just incredible collection of people from all over the world. And I've never met more passionate or committed people, and they're all committed to, making a difference in people's lives. So that's my story, and I'm sorry I didn't leave time for questions, but I definitely can take them if Kristen has them and send them to me electronically. Thanks.
Yeah, Dr. McKinley, Mark, thank you so much for your time and all of your insight into what you've done. My gosh, in one person, it's really incredible, and we're grateful that you came to share that with us today. And I will definitely forward any questions that come in. Thank you so much for that offer. And we just have a few comments coming in saying how incredible that was with you as a speaker.
Well, thank you very much. I really appreciate it. So are there any questions I can answer now, or?
Feel free to unmute. I know a bunch of people just turned off their cameras during the presentation. Okay, well, if you think of something, oh yes, Bruce.
I just have one comment that maybe you can then comment on. I was struck by, I think you said for polio, it was 1 in 200 people that got serious symptoms. Was that correct?
We've become paralyzed, right?
And then
yes.
To me, and not to go off on and become an anti-vaxxer at this moment, but folks who complain about having to get vaccinated for COVID when only 1 in 200 people don't recover for it. That number stuck out to me as how the lines for polio when so many people today are scared of getting vaccinated.
Well, no, and it's the global impact. I mean, that's the question that came up. If we eradicate polio, the amount of money that will be saved globally that's put into these vaccination campaigns is in the billions. I forget how many billions and billions it is. So it's a huge savings for everyone. So then we can focus on something else. So it's really critical to try to finish this job. Yes. Yep.
We had another question. Is there still no hope for Pleconaril?
So Pleconaril, as I mentioned, an investor bought the rights to the drug from Sanofi and is working to try to get the drug approved for neonatal enteroviral sepsis. So this is a virus that typically in the summer, late in early fall months, you'll see people with aseptic meningitis and or myocarditis, different serious diseases. And women who are pregnant, if they get infected, can have term delivery and the baby can be infected and it's lethal 50% of the time. And there's nothing you can use. And our drug reduced that mortality by 50%. So we're making the argument, okay, we've got a safe drug. We know how to deliver it. You know, come on, FDA, this is not the common cold. Let's go. And so I have to say, they've been very, the same person heads the division that was there when I pitched the cold. So she knows me very well. But yeah, they've been very helpful in trying to, you know, take care of all the little things that need to be taken care of to move it ahead. So hopefully it'll be approved.
Anybody else? All right. Well, I know whenever I go to the doctors, I think of the good questions afterwards. So
Oh,
anyone,
no, that's fine. That's fine. I
Anyone-
appreciate it.
well, thank you so much for your time. Again, it's fascinating. And I wish we could have 3 hours with you, but we appreciate the time you were able to give.
All right. Thank you all. Take care. And I'm not
Thank
seeing how
you.
to get out of here now.
Should
Um,
hover and then end. Or leave.
It's not
They leave.
the menu that allows me to not share my screen.
Up-
Okay.
up
Here,
at-
let me.
Up at the top, there should be a green.
All right. I'm just going to close the window here. All
Okay.
right. Thank you. Bye.
Thank you.
Bye.
Bye.
Oop, no, I'm still there.
Surprise.
All right, there we go. I helped him. All right. Okay. So thanks, you guys. If your camera's off and you want to turn it back on and join in, that'd be great. And if you feel more comfortable leaving it off for now, that's fine as well. Or if you're having bandwidth issues like I am, feel free to leave the camera off. But we will get right into today then. And Janet is joining us as our master instructor today. Hey, Janet.
Hey, so just really quickly, I'm going to get off my ear pods. Can everybody hear me okay?
Yep.
All right, so let's go ahead. We're going to do the qPCR lab today. Brittany isn't going to join us today because she unfortunately is sick. So I thought let's go ahead and just check and make sure that you have all the tubes that you need and all of the reagents that you need going ahead and thawing, if that's okay. So the first thing you're gonna need is 4 PCR tubes.
I'm going to put this in the chat as you say it, Janet. And I think that you might have volume.
Then you're gonna
on
need
your computer.
2 microcentrifuge tubes, empty ones. So again, microcentrifuge tubes. It would be helpful if you had a Sharpie or something to label your tubes. Now, in terms of the reagents that should have been in your frozen box, you need, it's got a green lid on it, and it should say DNA sample.
Well, you guys have the little
Oh, I'm
tube.
sorry. Yours probably don't have green lids. I'm sorry. Forgot about that. So it should say DNA sample. Then you should have nuclease-free water. You should have qPCR primers. And then lastly, you should have 2X P green master mix. So does everybody have these things, have any problems? Also, of course, you'll need your micropipetter and the tips. I always forget, but a beaker for waste tips is always useful. Okay. So are we ready? All right, those things can be thawing. It's okay that they're at room temperature because we're going to need to pipette them. So I'm going to go ahead and share my screen and go through the basics of what we're doing today. And if I can find it. So this is in the slide deck and it starts on slide 90. But basically what we're going to do today is be able to see the change in concentration of PCR product through fluorescence. So I'm going to go through this. This is when they do a PCR test, this is what they're doing. In 90% of the hospitals, they're using qPCR right now for those PCR tests for COVID tests. So you can then, by looking at the fluorescent readout, you can predict what the viral load of RNA was in the bloodstream or in that swab. So it's a really good quantitative method to be able to predict what the viral load is. So we're going to be then predicting the DNA concentration through an unknown example. And we're going to compare and contrast eventually the difference between endpoint PCR, which is normal PCR, and real-time PCR, which are the assays. So you can use, if you don't have, if you have to give back your blue gel because it's borrowed, you can use these, they're called P51s, and they're really inexpensive from Mini PCR. You can use them to do this lab as well. Okay, so what are we doing today? And again, I'm shorthanded in the room, so if there are questions, somebody like jump up and down and scream and yell and get me to stop. So I know for the last 2 weeks, you guys did the food safety and the genes in space, so you should be really well familiar with PCR. But again, we're starting with a small amount of DNA, and then we're getting to a large amount of DNA, many copies. In the case of course, COVID is an RNA virus. And so the only difference is that you're starting with an RNA sample and then measuring the RNA at the end. And so you can tell your students really the only difference is the DNA to RNA. All of the other processes are the same. So again, the PCR reaction takes place at 3 general temperature ranges. The first one is denaturation, which is when the double-stranded DNA, the hydrogen bond breaks and it's separated into single strands. And then we have the annealing step, which is where the temperature is lowered, where it is idealized for those primers to find their complementary base pairs and to align and anneal to them, which then allows the polymerase to attach to that primer site. And then during that 72-degree step, go ahead and build using those free nucleotides to continue the extension step. So obviously we do this in the thermocycler. Today we're going to be running, you can run it up to 30 cycles, but the cycles are timed so that they're much shorter. And you'll see when we get to priming it what I'm talking about. So again, in our reaction mixtures, you have your template DNA, you have your primers, polymerase, your free, the DNTPs, which are the denucleotide triphosphates, and then the buffer that's going to keep the proper pH concentrations so that we don't denature as those temperatures fluctuate so much. So now, obviously with PCR, you have the products increase exponentially, doubling every time. So the area to look at is here, this area here in the middle, where it's in that growth phase, and it's just increasing so rapidly. And then eventually it reaches a plateau because it's run out of reagents. And so then you don't get any more copies. So when we do a traditional PCR, we run it for the full 30 or 35 cycles, and then we run a gel afterwards. And so we only see that end point, how many copies we have at the very end. And by that point, it's impossible to extrapolate backwards to figure out what was the starting concentration, because we've so saturated. And so there's no way to go backwards. But if we were to somehow magically be able to look at this cycle, at this curve here, and on this graph, it's between 20 and 25 number of cycles, we would be able to maybe measure a difference and be able to use standards to be able to, like I said, extrapolate backwards to the original amount of DNA that was in there. And so if you look here at this graph, at the green, the gray, the blue, and the yellow, they all end at the same endpoint. But they all hit that endpoint at different times. And this is the essence, this is the crux of qPCR, is being able to quantify when it hits certain targets and know how many copies were made, and then be able to go backwards through just a little bit of math to quantify then at the beginning. And obviously, you know, for regular PCR, we just want endpoints, right? We only want to have nice bright bands. We don't want to stop it in the middle, because on a gel, we're not going to see that amount of DNA. It's going to be faint bands. It's not going to show up for us, and it would be a failure. But with qPCR, what happens, and here's the qPCR, is again, we're looking at this range. And so qPCR uses a fluorescence tag and thermocycling to be measure both at one time. Does that make sense? So you're measuring fluorescence and you're measuring, you're going through the PCR cycles both at the same time. So if you look at that amount of fluorescence, here's your target DNA. You start off in this case with 1 copy, and then it goes, as it's doubled, that fluorescent dye that only adheres to double-stranded DNA is going to be in a small amount, but then again is going to 2 to the n, right, for every cycle. So here we have 1, 2, 3, 4 cycles. This would be 2 to the 4th amount of fluorescent dye. And so qPCR machines are really expensive. They're tens of thousands of dollars because they're basically a fluorometer and a thermocycler at the same thing, which ends up being very expensive, unfortunately. But luckily for us, we can do it here using our little blue gel. So here, the amount of fluorescence for that double-stranded DNA will be minimal if you have just a small amount of DNA to begin with. But then as it starts increasing, there will be a point where it will start to fluoresce. And if you can catch it, for us, if you can catch it and pause it in the middle of the cycles, you can see that fluorescence and you can compare it to known samples. And then you can sort of guesstimate, well, does it have more or less DNA than our known samples? And so it's that threshold is known as CT, for cycle threshold. So basically what we're looking at is trying to figure out how many cycles did it take to get to the threshold where we can see it. So as I mentioned before, obviously the starting concentration matters. And if there's a twofold difference, right, in the amount of DNA, or excuse me, if there's a twofold difference in the number of cycles, then you expect a fourfold difference in the starting DNA. And that's again because it's 2 to the n, where the 2 is the doubling and the n is the number of cycles. So if we look at here at these two, and I know my screen is small, I'm going to try to blow it up for you without making it too blurry. Where'd it go? Okay. Let's see if I move. There we go. Does that help? So if you look here, if we were to estimate that this is at cycle 16, where we're able to see the fluorescence, and this is at cycle 18. So the green line here is 2 cycles away, which means then that it's got 4 times as much DNA as this cycle here at the green line. And I don't want to go too fast, so I'm going to pause here if you guys have questions, because it took me for a minute to figure out the math. First of all, I am not a math brain, and secondly, I'm dyslexic, so I reverse things all the time. I'm going to try to check the chat if there's anything going on. Let's see. Are we good for everybody?
I'll just say quickly, I've done this a number of times and the math trips me up every single time. And so in the lab, there is like a worksheet for kids to practice doing the doubling. I don't know if you were going to mention that, but that is a good, just
Yes,
to train your brain to think about exponential growth, because it is a little
Yes,
difficult to go back and forth.
that's an excellent point. So then again, you can see if we go then to the other reaction lines, that if you look at the blue line and you look at the gray line, a question you would ask the kids is which one was more concentrated? And obviously the gray, because it hit the threshold earlier. And then you would have to then do the math. So 2 times n, 2 to the n, where you calculate then if the gray line was at 18, 19, 20, 21, 22, the blue line is 4 cycles beyond. So 2 to the n. So then that would be 8 times as concentrated. So by comparing these PCR reactions in real time, you can calculate. And again, this is, I don't even know the number of tests that are now done through qPCR. And they even at most hospitals, they have what are called multiplex PCR, qPCRs, where you can put in a number of different, you can put in one sample and they will combine it with lots of different primer pairs, and they'll test for 8 or 10 different viruses or bacteria at one time doing qPCR. And so that's how you can often get the rapid tests. You don't have to wait the 24, 48 hours for culture tests anymore. They just do a qPCR and get you the lab results in a couple hours. Of course, not right now for COVID for most people, but it does exist for that. Okay, so what are we actually going to see? And this may be a little bit frustrating for your students. I had one of my lab aides actually run it today, and he told me he really couldn't see much of a difference. But then I pointed out that you really have to look at the difference in the standards. So you're going to run, and your students would run, a negative control and a positive control. And it's by comparing your experimental to that positive control and that negative control where you can actually see the difference. So if you look here at these 3 tubes, you can obviously see a difference. Sometimes students aren't, their eyes aren't tuned to look for that amount of detail. They'll just look at them and say, oh, they're fluorescing, and they don't see that actual detail. So you need to make sure that you point out to them to compare it to the unknown, or to compare it to your controls. Okay, so the first thing I want you to do, or the next thing I'd like you to do, is to go ahead and turn in your books, in your lab going viral binder, to the section for qPCR, and it's page 12 of the qPCR. It looks like the laboratory guide. And so you have to decide at this point, how much do you want to dilute your sample by? And I'm shortening, actually, I'm going to pause for a second. Would you prefer if we went through the pre-lab process, if you could show me on the chat what you would like to do? If you want to walk through the pre-lab that you would do with your students, or if you want to jump into the lab. The lab does take a little over, I think, an hour to run. So I wanted to be cognizant of our time. Of course, this lab you can finish up on your own if we go over time, and then just throw the tubes in the freezer, and we can, we'll run our gels next week. So can we just give me feedback in the chat, what you would prefer to do? That would be helpful. And the question is, do you want to go over the students pre-lab, or do you want to jump into pipetting? And I can tell I've put everybody to sleep, which seems to be common in my classroom as well.
Not true. You're amazing. Everyone's probably taking notes or something.
Okay,
We have
let's
two
go
votes for
through,
pre-lab.
then the pre-lab material. So if you look back then in the lab notebook, they go over a lot of that background material that I just flew through. And so it goes through, obviously I would not start off with qPCR. I think it's a more advanced skill. I think students would really need to have a background knowledge in what PCR is. For example, I'm going to do qPCR with my second level of applications of biotech class. I'm probably not going to do it with my first level of essentials, just because for lots of reasons, but it's nice to be able to save a lab that's like special for applications, but also applications. By now, I don't have to remind them what PCR is and how it works, and cycle numbers and all of that. There is a comment. So sometimes in the media or in science journals, you'll see the term RT PCR. And RT-PCR can actually mean two different things. So RT-PCR used to be the shorthand for real-time PCR, which is qPCR, which is that quantitative PCR. That's what the Q stands for. But now there's a process called reverse transcriptase PCR, which is basically when you go from RNA backwards to DNA and then use it. And so when you use RT-PCR, now it's hard to tell whether you're using real-time PCR or reverse transcriptase PCR. So if you get into the habit of calling it qPCR, then you avoid all that RT-PCR nonsense confusion. And then if we look at that page 12, so let's walk through how to decide what dilutions you want to make. So you are starting off in your tubes in your reaction DNA tubes. That DNA is given to you at 40 picograms per microliter. So that is 40 picograms. I'll write it for you. So just so you can see, so that's 40 grams per picoliter, and that is your known, that's your positive control. Now, the purpose of the experiment is to be able to figure out, you know, be able to quantify what are you starting with, be able to use when you can see it fluorescing through the PCR cycles, and be able to work back and verify that was the concentration. So you need to decide what concentration you want to dilute this to. You have 20 microliters of your starting DNA. You do not want to use more than 10 microliters of that. You need 10 microliters for your positive control. So for simple sanity check for me, I started off with half. So I took of that 20 microliters I had, and I'll show you the math. I did a serial dilution. So I did, and I'll hold up my serial dilution. That's on the next page. Let's see. And I'll talk through that. In the first tube, okay, I put 10 microliters of my sample DNA. And you can go ahead and start pipetting if you want to, or you can wait for a minute or two and I'll pause and let you do your pipetting. And then I added 10 microliters of water. So in essence, I halved the concentration. So in my first experimental tube, I had 20 picograms per microliter. Then I wanted to run a couple more tubes. And if you are working alone today, you're going to want to make an unknown, a second tube, experimental tube. If we're doing this in the lab, the students would be trading with each other to actually figure out the concentrations. But here today, I would just go ahead and do another dilution, half dilution. So if you take, now I've got two tubes. Right, where are my tubes? I have my E1, which is my original amount of DNA. Then I've taken 10 microliters out of that, and I've put 10 microliters of the sample DNA into tube 2. And then I've added 10 microliters of water to that. So now I have my positive control and I have experimental 1. Then I would take 10 microliters out of experimental 1 and put it into another tube, which I called E2. And so to that 10 microliters that I added into my tube, you put another 10 microliters of water, which again, halves the concentration from this second tube. So basically when you look at the tubes, I have full strength, half strength, and a quarter strength. Questions about those? If you've never done a serial dilution, this can be really confusing, but it's actually really easy. I say, I tell my kids, do serial murderers change the way they kill people? No, they kill people the same way every time. So a serial dilution is doing the same thing every time. And that's probably a really bad thing to say. I'm probably encouraging people to go be serial murderers. But anyway, so you take the 10 microliters from your first, put it into your second, add 10 microliters of water. Take 10 microliters of that solution, put it into the next tube, add 10 microliters of water. So now that you've done that, and I'm going to pause for a second. So if you haven't done it, go ahead and pipette it, because it's these experimental tubes and your control tube that we're going to be using in setting up the qPCR. Okay, so we have our E tube. And we have, I called it, when I made my dilution, I labeled it E2, but you should label it. So if you look on step 4, you've labeled your tube E. That was your first dilution. And now for step 6, you're going to label that new tube. And this is where you're going to be, your second dilution will be if you're working by yourself. If you're working with people together, then just go ahead and swap your samples if you want to. This is where the kids would just trade their U-tubes. That's funny. They would trade their U-tubes. And then go ahead and turn on page 14 and look at how to set up the PCR tubes. And if you want to, go ahead and unmute yourselves so you can yell questions. I'm working blind by myself today. Like I said, I have my phone up for one and I have my laptop up.
I have a question.
Hey, Brandy.
Hey, so what am I starting with? Like, is it this 20, I've got 220 TBE, or is this supposed to be pulling out of the freezer?
Can you say that again? I missed part of it. Go ahead.
What do I start with?
Oh, you should be starting with what is called your DNA sample. So did you pull those from the freezer? I'm on.
Let me go get that. Sorry, I lost the internet connection at the school.
Oh,
So
I'm so sorry. Go ahead.
you're gonna miss that piece.
So how about everybody else? Are we all good? So you're going to be starting with your sample DNA.
So I just want to clarify, we are adding 10 microliters of our nuclease-free water
Yes.
to the DNA sample tube.
No. Good question. You're
Okay.
going to take that DNA sample tube.
Mhm,
You're going to create a new microcentrifuge tube,
mhm.
and you're going to label that E.
Okay. Isn't there only 10 microliters in here though?
There should be 20.
Okay.
If there isn't, we'll adjust our numbers. But I'm pretty sure we gave you 20 of the sample.
Okay.
Oh, and I want to call out Megan and Kristen. Remind me, who posted the sweet pictures of their kids doing the micropipetting lab? Was that
It's Felicia.
Felicia? Your kids are adorable. I loved seeing their sweet faces.
Everybody did. I sent it around to the board. And that's like the best response we've had. They were so excited to see that.
Yes, because it's all about the kids.
It was awesome.
So Brandy, did you find your tubes?
I have the DNA sample
Okay, great.
and I'm pulling 10 out of it and putting into a new tube labeled E, but
Correct.
it seems like this is empty now. So
Is it? Well,
I agree with the other lady that I think it's only 10.
okay, well then let's pull 5.
Okay.
5 and 5 will work just fine.
Yeah.
I mean, I did have 10. I had 20 in mine
Okay.
where I pulled it.
If you need to, you can flick your tubes down, just with your wrist to try to collect the liquid at the bottom if it's redistributed. Or of course, if you have a fancy little centrifuge, you can do it with a centrifuge. Bruce, are you from Mini PCR?
Yes.
Oh, great. Well, I'm sorry, we didn't get a chance to introduce you.
Yeah,
Or
no
maybe you
worries.
missed that.
Yeah, hi, I'm the director of curriculum at Mini PCR. So I know this lab quite well.
I'm sure
happy to
you
answer any
do.
questions.
Bruce did last week's lesson.
Oh, I'm
Week
sorry.
before, yeah.
I'm sorry.
No worries.
I've watched, done them several times when you've done webinars.
I forgot to put on my Mini PCR attire today, so I'm
We'll
hard
forgive
to
you.
recognize.
We'll forgive you. So I've added on my screen an example of what the concentrations would be if we did what we described. So if we halved the concentration from our reference tube, that would give us 20 picograms per microliter in our experimental. And then if you did that again, halved it again, for your unknown, that would give us 10 picograms per microliter. Brandy, how you holding up?
Good.
Okay, is everybody else caught up with us? All right, great. So now in the classroom, of course, the students wouldn't know what the concentration is of the U, and you wouldn't tell them how to make the dilutions. You would have them figure something out. And I would actually lead them through and have them really think critically about the math behind it. I would have them do some doubling, exponential practice to have them think critically about what would be a good dilution to try out. I wanted us to have a sample and just go quickly into this. So now if we get to your PCR tubes, which I put mine down someplace, there they are. You should label your PCR tubes 1, 2, 3, and 4. And I see that they're recommending to label here along the side. I don't see where, here up in this region. On my large thermocycler, not the mini PCR, but on my large thermocycler, sometimes the ink comes off if I label it on the side. So I also have to label, I often label up in this ridged like little area up in here, where you will also see there are numbers up there already. So sometimes the heat cooling cycle picks up ink for whatever reason. So I have had students go, oh my gosh, we have no idea whose tubes are whose. So that's just a little hint. So you've labeled 1, 2, 3, 4, and then you're just going to go ahead and pipette exactly like we have here on the chart. And remember for tube 2, that R, that's your positive control, and that's the sample, the DNA sample we sent you. Tube 3 is your experimental, which would be your first dilution, and tube U is your second dilution or an unknown from a friend of yours that's in the room with you. Hopefully wearing masks and socially distanced.
So if we changed it to 10, to 5 rather than 10,
Yes,
what do we use here?
I would go ahead and use all 5 microliters. Bruce, would you agree with that? Yeah.
He's muted,
Sorry, yeah,
I saw.
yeah, it's asking for 7. Wait, where is it?
7 and a
Template DNA,
half.
7.5. So if you only have 5, that'll be fine. It might take a smidgen longer than a cycle longer, but you'll still see differences between your tubes. And
But if
when you
you-
put the 5 and 5 together, you should have enough for the 7.5.
I was going to say, if you put 5 microliters of the DNA in and then added 5 microliters of the nuclease-free water, you should have 10 microliters total.
It would be in the original
but in
tube,
the, yeah,
if
it,
they
you're gonna
didn't
have to
have
go back
enough.
to that original tube. That was my
but
bad.
it'll be all right. In this like, just to be really clear, in qPCR you can get very, very precise. In this lab, you're going to get generally more and less, and that will still be true if you're a little off on the volumes.
How low can we go for the master mix? Because I only have 15 microliters.
Of the master mix?
Mm-hmm.
Um,
It down and it's all I have.
total?
Yeah,
Is there another tube of master mix?
no. So I didn't know if there was, I didn't know, Bruce, if there was a level I could go to that would still give us decent results.
No, the master mix, you need what you need there.
Okay.
So you have, it calls for 15. Is it a 30?
Yeah,
Yeah. If you only have 15, that's kind of unfortunate, but I would set up, if you can only set up, that's enough to set up one reaction. I would just set up your experimental reaction and you will,
Okay.
you'll still see the tube go, you'll see one tube go from boring to exciting.
Cool.
You won't see the difference though, between tubes.
Will you let me know if anybody else had that problem so I can grade my lab aid?
Oh, yeah, Laura said that. Hmm.
Okay. All right, and then of course, in a PCR reaction, we know all the good things that are in there. I'm pausing while people are pipetting. And if you are done pipetting, you want to go ahead and get your mini PCR and, you know, plug it in and get it ready to go. And you also want to get your blue gel set up with your holder ready to go as well, so that you can measure, you can take your tubes out of the mini PCR and put it right into the blue gel to visualize right away.
Miss Janet.
Yes.
So what is the primer? So, okay, I've got my master mix. I found it in my tinfoil. Is that what is labeled ladder or dye or XMNL?
No.
No.
That is the XMNL is a restriction digest or a restriction enzyme. It should be called, it should be labeled qPCR primers.
Okay.
And again, I have learned that I will, if I get to do this again, I will make sure that the tubes are color coded and I know the color coding so that I can point people to the right color coded tube. The things you learn.
We will label them several ways.
Yes. And maybe next time we won't have to do it virtually. Maybe we can
Right?
be in the same room. Sorry, I'm whiny.
Good news is, what, now 8 more wake ups in the early morning until break. If everyone's last day is the 18th.
Oh, you guys stink. We're going through the 22nd.
Sorry.
That's not fair. Okay, so are we ready to go ahead and move on to the setting up the PCR settings? Okay, so the first thing you want to do is take your tubes. I'm going to go turn the light off. I have not pipetted anything. I just am not that coordinated to talk and pipette at the same time reliably. But I have a couple tubes that were done earlier today. So you're going to put it in and visualize. You should see and be able to record fluorescence or not fluorescence and kind of score it. Oops, and not drop it on your laptop like I just did. And so you want to scale them just like it says here and scale that brightest is the 3, and that's your comparison. I encourage my students when we've done this to go ahead and take a picture on their phones so they have that comparison that they can pull up side by side eventually and compare the color, the fluorescence, not the color, the fluorescence too. And then the students go ahead and record that in table 1. I see Felicia's in the dark over there. Anybody having any issues getting there? I know it's hard to run and go ahead and get the lights and then turn the lights back on and all of that.
So Laura said her primer tube was empty and I asked her to shake it down. I think she found a little bit and she said if she has to choose which of the 4 is the most important. So we said the experimental tube, correct?
Yeah,
Yes, okay.
because she can compare it against other people's things. Excuse me, this is all kinds of weirdness is going on. My mom's in the hospital, and so I just have to take a minute to answer this. I am so sorry.
That's okay.
Yeah, I think everybody needs a minute to pipette and reset, so.
I can jump in for a second too. What's your,
No, I got it. It was
Oh.
just a text message. Luckily, it's not COVID, but so it looks like lights are off for the people I can see. So I'm going to go ahead and share my screen. I'm going to plug in my thermocycler, but your screen should be like this. Notice that the cycles are really short, which surprised me the first time I ran this. I was like, 8 seconds. That's like so short. How does it even do that? But it works. Makes me think that maybe I can shorten the cycle on some of my other PCRs. So let's see if I can get to my.
most PCRs are a little over-engineered. That's one thing that we found.
Yeah. Yeah. So are you guys setting up your PCRs? If I go to my Google Chrome. Now I've lost you guys.
We're here.
Oh, I give up. I'm not going to give up. So there we go. I found you.
Yay.
So I'm sharing my screen. Okay, so I'm hoping everybody can see my screen. So I'm going to go ahead and open the qPCR, which I already loaded today. If you don't, obviously you probably don't have that qPCR, you just hit the add button and you can add a new protocol. And so go ahead and name it qPCR, and then you're going to set it for the settings that are on this other screen. So there are the settings that you need. 94 for 60 seconds, 94 for 8, and then keep going. So can you guys give me a thumbs up or something when you're ready? Your tubes are in the thermocycler and it's all programmed and ready to go. So are we ready?
Ready.
I'm waiting. How are we doing?
Ready to go. If we're ready, do you want us to go ahead and start?
Okay, that's great. Oops. My laptop's about to die, so I have to get another thing going. If anybody wants to take over my life, I will gladly turn it over to somebody. Okay, so now the next thing you want to do is go ahead and put your tubes in and watch your timing on the number of cycles. So once you've got it running, so I'm going to pull this up and you hit run, you want to, I'm going to connect to my machine. I'm going to turn my machine on. Sorry, I should have been doing this. So here's my mini PCR screen. And what you want to do is about 30 seconds into that initial denaturation, you want to pause it and grab the tubes and put it in your blue gel to be able to visualize it. You want to make sure that you don't touch the metal parts because they obviously get really hot. So is everybody clear on that? You're going to go ahead and start it and you're going to pause it. Then I would say try to hit it somewhere between 30 and 45 seconds and then go ahead and view it on the blue gel.
When you hit pause, just so folks know, it stays at the temperature that it's at, or it continues going to the temperature it's going to. So if you realize you press it too early, just wait and it will stay at that temperature. So it's better to hit it a little early and just wait a little bit longer and then look. If you wait too long, it'll go past it and it'll start changing temperatures. That's all. And you don't want to touch the metal, but I've touched it plenty of times. It doesn't burn you very badly.
I like that very badly.
Yeah, Janet, I think we can't hear you talking. Can other people hear Janet? Can Janet hear us?
She can hear us, yeah, I think. Still can't hear you, Janet. Still no.
Well, I've been amusing. Whoa. I've been amusing myself. For what, 15 minutes? I've been talking to myself. No wonder nobody answered me.
There's only a
This
couple
is not
minutes.
my day, you guys. I am so sorry. You'd think by this point I would know what the heck I'm doing. So at this point, you want to catch it at that 10 cycles. Oh my God. So a question that I had asked is, why do we want to pause it during the extension phase? Which nobody had answered me. And I was like, wow, they're really ghosting me. But now I know why. So why do we pause it during the extension stage? Well, I will tell you. So this is a great question for students to see what their understanding is. So again, remember that fluorescent molecule binds to double-stranded DNA. And so in the extension stage, that's where your polymerase is extending and making double-stranded DNA. And hopefully when you catch it at the end of the cycle, it will have gotten, if you think of it being a single-stranded DNA and that polymerase is going along, you will have gotten to, that polymerase will have reached the end, and you're going to have a large strand of double-stranded DNA. So if you do it, of course, if you pause it during and try to view it during the denaturation, that 94 degrees, you're going to be looking at single-stranded DNA and it's not going to fluoresce. And you can pull it and check it. And that's something I would ask the students to do. If they doubt me, go ahead and pull it during these different temperatures. And of course, if you grab it and pull it at that 55 degrees or 54 degrees, excuse me, during the annealing stage, that's where your primers are attaching. What are your primers attached to? They attach to single-stranded DNA. And so I don't know where we are in our timing of that 10 cycles, but go ahead if you've check your thermocycler reactions and see where you are in terms of how many cycles it's gone through. How are we doing?
I'm on 5 right now.
You're at 5? Okay, so what we're going to do this week is when your cycles, we're going to pull them at 10, look at our tubes and kind of talk through what we've seen. And then I'm going to encourage you to go ahead and, if you can, leave your PCR running to finish the 30 cycles. And if you do that, then we can run these on a gel at the beginning of the week next week. Next week, we've set our time to be really our sharing session where we look and plan lessons, work together on figure out and talk through curriculum and look at where some of these lessons might fit in, share ideas with each other, talk calendars. But I'd like to start off by running the PCR products from today so you can see on the gel as well, what we saw as the result of the qPCR, but at the end point. Does that make sense to all of you? Great. Thank you for the visual feedback. That really helps. The other thing I wanted to ask, which I had asked and Bruce ghosted me, but I realized now I was on mute. So Bruce, I wanted to ask you a little bit about the curriculum development from Mini PCR. By the way, your stuff is fantastic. I'm a connoisseur of lots of different companies, educational materials, Biorads, Edvotex, Mini PCRs. You guys are by far, I think, some of the clearest that's out there.
Thank you. Glad you like it.
So are you guys working on new things?
Oh, we are always working on new things. Yes, that's what we do. Yeah, we have a curriculum team of 4 or 5 of us. We actually just released, or it's in that we've announced, haven't quite released yet, I'm not sure if you guys have what you have done, but a CRISPR activity where you can do CRISPR in bacteria. If I believe you've played with the BioBits things, we have a new BioBits lab coming out. We also have a new lab that I've previewed, I think you mentioned one of those webinars, based on a collaboration that we did with the Duke Lemur Center, sort of showcasing their work in an electrophoresis lab. But yes, we're always trying to think of new things. And I don't know, I don't remember how I introduced myself previously, but I'm a formal high school bio teacher and taught AP Bio and taught all sorts of different levels. So I'm glad you like the materials. I try to make it something I would have wanted to use in my classroom. And that's sort of like the standard that we try to, if we think it would have been useful to us, we put it out there in the world. So.
Janet, we can't hear you again. Oh no, Janet lost her voice. Janet, no. I saw you unmute, but no. Is it connected to your AirPods?
Well, I'll give a fun little anecdote about this, and I don't think you guys did the enzyme activity. But those two activities, this one was actually originally largely, a lot of it was developed by an intern that I had, who was my AP biology student. So it was done by an AP bio student. He went to college, and after his freshman year of college, I had quit my job and taken this, and he came back and worked for me. So this was one of the first developments of one of my former students. So it holds a little bit of a place in my heart as well.
That's awesome. Full circle. They're coming back.
Exactly.
Now Janet has gone
She left.
gone videoless. She's disappearing piece by piece.
So just, I don't know if maybe if there's anything you want to throw into the chat, Janet, or dial in through your phone. Let me look that up.
The phone's dead. Cool.
So I'll preview what folks are going to see here. I'll jump in for a little bit. So the key thing that you want to, the lab asks you to check it at cycle 10, I think, or maybe 12, I forget which. You'll probably see, and then it asks you to wait every 3 cycles and just look if things are fluorescing. What you're going to see is it varies just a little bit based on pipetting and error and whatnot, but usually somewhere around cycle 15, you'll start to see your tubes glowing, your first tube glowing. And then if we doubled it, or if we cut it in half rather, what we did here, it'll be every few more cycles, another tube will start glowing. And from the first time you see it, it'll be very faint, and you'll compare it to the negative that you have. And the next time you look at it, it should be a little bit brighter. And usually by the 3rd time you look at it, it's super, super bright. And it's just very clear that it's glowing. So the first time you see it, you'll look at it and you'll be like, I'm not sure if that's fluorescing differently or not. And if you have a practiced eye, you'll pick it out a lot quicker. And then as it comes, it'll get brighter and brighter. And that's just a great way too in this lab. As Janet said, I wouldn't do this as the first PCR lab that people do, but it definitely reinforces that concept of how PCR works at exponential growth. It starts out, it'll just barely be there, and then really quickly it shoots up to the top. And then the next one, you'll just barely see it, and a few cycles later, it'll be very bright.
So thank you, Bruce, for jumping in there during my technical difficulties. Every piece of appliance I have is losing battery life at this point.
The end of the day.
Oh my gosh. So how are the tubes looking? Are you getting to see anything different? Are you seeing any results?
And again, it'll usually be about, something's glowing there.
Nice.
Yay.
There we go. It's nice and bright.
Yay, so we have a
Ooh.
working tube with everything in it.
Yay.
Yeah, and it all depends on the temperature that you look at it too. They're always going to, at low temperatures, this is one thing why it's good to look at it when you start. If you look at a root temperature, it's always going to fluoresce just because it's cold enough that those primers, stuff in there, even if they're not supposed to bind together, they're going to find something to stick to because it's cold. So at cold temperatures, it will always fluoresce. At hot temperatures, it will never fluoresce because you've cooked it. And so it's that sweet spot at that 72 when it's just finished. And this is what Jan was saying earlier, when it's just finished. End of the day. Replicating.
Extension.
Thank you.
Oh no, Bruce's brain is going too.
Yeah, I know. I work at a company
I'm
called
rubbing
Mini
off on
PCR. It would be good if I know the steps of PCR. Yeah, right at the end of that, when you've made the 2 strands, there's nothing else in the tube bound together, but you can see that double-strand DNA glow.
So while those are running and we're kind of finishing up, I'd encourage you to look at the page 22 which is where the students would do the concentration calculations. So it looks like that.
Not really a lot.
Yeah, I couldn't see
What?
much.
We're seeing like a little
Oh.
window. Yeah.
Sorry,
I don't know. 22 though.
page 22. If you take a look at that, the students on the previous page have recorded what they've seen for all of the tubes through all their different time points. And again, what I really like is that they choose what time points they want to measure based on what they think and hypothesize what they'll be able to see. And then on the page 22, when they're doing the calculations, you're having them look at where was that critical threshold, right? Where was that first time that they actually started to see fluorescence? And so what cycle was that? So in tube 1, obviously that's your negative control, and that should be nothing. They should not see anything fluorescing in their water tube. And if they do, they pipetted incorrectly. For tube 2, that's your reference DNA. And of course, that is your strongest concentration because that's the source of all of our DNA. And so you would think that that critical threshold, you hope that that was much earlier in the cycles, in cycle numbers, I'm sorry. And then tube 3 is your experimental, and looking at when it started to fluoresce. And then tube 4 is the unknown. And of course, tube 3 and 4, truly with the students, when there's unknowns, tube 4 might be fluorescing earlier or tube 3 might. And so when they go to look at those concentrations, if it's your reference tube is 40 picograms per microliter, that's basically that concentration is 1x. And so then if you look at the experimental, what did anybody start to see their experimental fluoresce after cycle 10? Did anybody see any fluorescence after cycle 10? Doesn't look like it. So then, you know, you would need to continue measuring at 15. So you're going to put, let's say tube 2 started to fluoresce at cycle 10, as an example. And then tube 3, your experimental, started to fluoresce after cycle 15. That is a difference of 5 cycles. So then it would be 2 to the 5th difference in concentration. And please don't ask me to do math at this point. Half my brain's in the hospital. So at that 2 to the 5th, then they can extrapolate back. So if 2 is 40 picograms per microliter, then what would be the concentration of that working backwards? Sorry, from the, I'm losing it. Working backwards, you can figure out then the concentration of the starting unknown. Whew, that was difficult. So at this point, I'm sure there are a lot of questions, or if you want to go back over any of the labs or discuss where you think this might fit in, please feel to like unmute yourselves and jump in. Or is everybody like I am with their brains on frozen dead batteries?
Okay, you said you use it in the application class, like about where in the class? You use it? I haven't
Yeah.
taught that class yet, so I'm probably will.
So Yeah, I'll teach it in my applications. I didn't this year at the beginning of the year because we're doing this DNA barcoding unit. But normally I would put it at the beginning of the year because it goes over a more advanced PCR technique. Because then in the spring, the students pick their own research projects. And if they wanted to do some type of qPCR, this might be a way that they could investigate.
Janet, at some point, do you mind sharing your kind of entire curriculum for essential applications? Because I teach out of the BioRad book, and you know what, I do what I can, but I have never been able to find something that would kind of like lead me down a tangible path.
I will share, I will admit that I am not very organized. So quit laughing, Megan. So my units, Brittany, we're the yin and yang. She's the super organized one. I'm like the creative fly by the seat of my pants kind of person. So I'll be happy to share my curriculum map. It has totally changed this year because of COVID, but normally in the fall for my applications class, my kids are really working through a synthetic biology cloning workflow. So they're extracting DNA or rehydrating DNA parts and then assembling them into plasmids and then measuring some output in the plasmids. And I use a combination of BioBuilder kits that are available through Carolina. The one I really like, there's two that I've used. The one I really like is called iTunes, where basically you're switching a promoter and a ribosomal binding site, and they have 9 different combinations, and the students can see the effect of promoter strength and the effect of ribosomal binding strength on an outputted color. And all you need is a spectrophotometer for that. And it works pretty well. And I have sample data that I can share easily to compare that to. I do like the BioBuilder because it talks about, I have an iGEM class, and I run an iGEM team, which is synthetic biology. And so for that, the BioBuilder helps prepare the students if they want to try out for my iGEM team, because it goes much more into depth in how to actually control cell output and protein expression and different things to manipulate. This year I'm doing it because of COVID. I didn't think I was going to have lab time. I'm doing a DNA barcoding that I got from Cold Spring Harbor. That's actually been fantastic. And I can share all of those materials as well. And Cold Spring Harbor has money, grant money. So that if you do this DNA barcoding, they can help support you with reagents to input it. And then they just ask that you share the DNA sequences and add to the database. And that my kids have really liked it because they got to collect insects and plants and develop their own hypotheses. They're actually doing their research experiments this fall for my applications class, because this was something that they could potentially do at home. And then in the spring, we're going to do the full cloning workflow and do some of that, like I said, that BioBuilder experience.
And I'll
So
vouch for the BioBuilder stuff. It's great.
How are our PCRs looking? I see we're dropping people off the call. Kristen and Megan, do we normally go to, I'm sorry, I just teach and I don't pay attention to timing. Do we usually go to 6:15?
We do. I think Laura Lambert got booted.
Oh, okay.
Yeah, I haven't seen Wayne County in a bit.
So yeah, I can put, if Laura's not on, but for any of you, I'll put my curriculum maps, both for last year when it was supposed to be normal and for this year, the COVID adjusted one, on the Google Drive. I'll clean them up a little bit and then I'll put them on the Google Drive, the shared drive, so you guys can see how I structure it. And oh, Laura's back. Yay.
Sorry, my computer died.
No, hey.
Last thing I heard was BioBuilder.
Oh my gosh.
Well, it's recorded. You can watch the part you missed.
Yeah, so I'll be happy to share my curriculum maps with you guys. And then there's a lot of materials that are like from Cold Spring Harbor that are open source, and BioBuilder that's open source, which is awesome. And at some point, I hope, I can talk more about what iGEM is, because it's a phenomenal program. And it's, for me, the single most impactful thing that I've done in my teaching. And I've been in teaching now 24 years. And I've sponsored a team for 7 years. And so the kids getting the chance to do their own research and then also work as a team, where science fair competitions are often, single students or just, 2 students in a group, 2 or 3 students in a group. This is a team. So they basically have to set up like and function like a biotech company. And then they compete as that team, and the dynamics and that collaboration are phenomenal. And it's open source. So it encourages collaborations between teams. So my team was on Zoom calls all summer and all fall with collegiate teams from around the world. So anyway, I won't proselytize on iGEM, but
Well,
I'll be
and
happy to
Janet-
share it. I'll put that all in the Google Drive.
Janet, I can share your webinar from this summer that you did
Oh,
with InFate.
Okay.
So yeah, since you talked about it for an hour, they can listen to that.
Yeah, they'll go to sleep after the first 5 minutes.
Nuh-uh.
Uh-huh.
Mm-mm.
So for next week,
Yes.
in order to start with running the gel, what does everybody need to have?
Everybody needs to make their gel. Do you guys remember how to make your gels from last week? So you take that that tablet you got in the mail, and go ahead. You can make a 2% gel. We'll work with a 2% gel. So do the same thing you did last week, which was dissolve that in 20 milliliters of water. I don't have the instructions in front of me. And then go ahead and make the gel, put the comb in, and have your buffer. And then we'll be ready to go ahead and run it. We'll load the samples along with, next week, you're going to need the ladder, which should be labeled ladder, DNA ladder, and you're going to need the gel loading die. And then please make sure next week that you have, access to curriculum maps for normal years, any of your standards that you need to cover. And next week, I hope to talk very little, and I hope everybody else gets to really talk and share, because I know I'm always looking for new ideas and what to do to incorporate. And I know you guys are awesome, else you wouldn't be in the class.
Probably one of the things I kind of imagine next week working as is that either leave your mute on or off, whatever you want, but just kind of talking into the microphone when you're ready to ask a question, or the light bulb goes off or something like that. Even if you're just thinking, okay, well, I'm thinking about putting it in February when I start X unit or something like that, and then just wait for some feedback, and that hopefully this collaboration with all of us together will be able to fuel some good ideas.
Yeah, so I'll share my curriculum maps by next week on that Google Drive for essentials and applications. And then I haven't taught AP Bio, but I'm AP Bio certified, but I'll talk to my AP Bio teacher and maybe get an idea of where he's fits things in, so that we can help whoever with some ideas.
And I'll just say quickly, I put my email in the chat there. So if anyone has any technical questions, especially, just feel free to email me directly about anything.
Thank you so much, Bruce, for joining in.
My pleasure.
Yes, thank you.
And we missed a webinar. That's the other thing is, guys, watch for the mini PCRs, get on their email list so you get notified on their webinars. Today, there was a webinar on using PCR for sleep studies, correct?
Yeah, our sleep lab. Yep. And those are
Yeah.
all just on our YouTube channel. There's actually, there's a webinar about this lab, using this lab to teach about COVID.
Yeah. Okay.
And
So
then
hang in
Laura,
there, guys.
Laura put a message in the chat too, that Bio-Rad is a good resource for PCR too. Or
Yeah.
All right. Thanks, everybody. Thanks, Janet.
Thank you,
Thanks,
everybody.
Bruce. Thank you to all the teachers for joining. It's such a long day, but thanks for pushing through. And we'll see you Monday.
Thank you,
All right,
guys. Bye.
Bye.
Y'all. See you.